Alright, hi online class. We just watched these two YouTube videos, and now Casey is going to explain stuff to us.
I don't know about that.
Can you be over here so you
Yeah.
can be recorded? Thanks.
How far over here? Okay. So I did the 4 properties of muscle tissue, which are I can't ever say it. Contractility, excitability, extensibility, and elasticity. And the way that you can remember that is by doing, can Ella eat eggs? Because it's the CEEE system. So that's all I had, but.
You actually, thank you.
Yep.
You anticipated where we're going today, which is great. Come on, little computer. We're on to muscles, which, in my personal opinion, is where it actually gets interesting. Like, bones are nice, and they're alive, and the anatomical tissue types are fine, but it gets more interesting when, like, it's more interesting today. Because stuff's doing things. Things are moving, they're doing stuff. Okay, so muscle tissue is all over the place, right? We're solved on this. And its big purpose is to take chemical energy and change that so you can actually exert force. So it's going to be snarfing down ATP like nobody's business. It's going to be going through this stuff dramatically quickly, but it's going to be exerting force. Yeah? Now, when I think of muscles, I think of like muscles that I use to, if I were theoretically to do a push-up, right? Like skeletal muscles. But you also have like cardiac muscle that's just chugging along just in general, right? You've got the muscle of your gut, you've got smooth muscle that's doing stuff. And even your skeletal muscles, even if you're just like sitting quietly, are still doing things. They're still like keeping you up. Maintaining posture is something you don't even really think about, but like the first time I saw somebody with a neurological problem that couldn't do that well, it was weird because like you don't even think about the importance of like keeping your balance and not being mush, but your muscles are going to do that. 3 types: skeletal, cardiac, smooth. You'll hear things referred to as muscle fibers. That's, yes, muscle tissue, but it's only skeletal and smooth muscle cells. Cardiac muscles aren't called fibers because they don't have that same shape. Just for what it's worth. When we see skeletal muscles specifically, it's a weird looking cell. Like, it is a very weird cell. The reason it's weird is, so both skeletal and smooth, they're really, really long compared to basically everything else. They're this really long cell. And they can shorten, they can contract, right? Like she said, they've got contractility. And for terms, every time you see this myo, or sometimes mice, MYS, you can think muscle, right? So we're going to see myosin, it's going to show up as muscle. Anytime you see this sarco, it means flesh. So again, muscle. You'll hear like with, we'll get to a bunch of sarco things. Okay, so far we're good? Oh, don't let me forget, as you leave, there are cookies. I'm so excited. So you should take a little baggie of cookies if you want to. They have peanuts, eggs, sugar, and vanilla, and salt in them. That's it. So, if that's a problem for anybody. Okay, great. Skeletal muscles. Attached to tendon by bones. They've got lots of nuclei. They're multinucleate. This is weird, right? Everyone's okay with this? It's weird. So, how come they're not more than one cell? If they've got lots of nuclei, if they're multinucleate, how come they're not just lots of cells? Like what makes it so that it's one cell as opposed to lots of cells just close to each other? Because they're long, yeah. There's one plasma membrane that goes around the whole edge, right? It's definitely one cell. It's really, really long. And because it's so long, you need lots. You need all these nuclei to be all around there because you're going to be making proteins, and you're going to be needing the DNA from a whole huge length of things. Muscle fibers are bizarrely long. All right, and there are other cells that are long. Like there's some neurons that are really long. Some neurons that like are in your spinal cord but go all the way down to your toe, like a sensory neuron. But muscle fibers, they're the ones that get lots of nuclei. Okay, we're good. But definitely one cell. There's definitely an inside, outside, the edge. We'll see it. It's going to be good. They also, we're still in skeletal muscle. They also look stripy. It's called striations. You saw it in your tissue lab, right? Looks stripy. And they're voluntary. They're subject to conscious control. All right. So yes, your muscles can react. They can like twitch. They can have involuntary reactions. But you can think about it and make them do things. We're good? Terms, so many terms. But you see all this mycium, muscle, right? So muscle tissue, it's got these muscle cells, right? And they're wrapped up in the same way that if you've ever seen like a really, really big bridge, have you ever walked across a huge bridge? You see those cables, right? Where there's like a wire, and then a whole bunch of wires, and they're wrapped around with wire, wrapped around, out and out and out into this huge cable. It's a really similar picture. We'll actually see the picture. See the picture, okay? We've got this muscle fiber, which is a cell, and it is freakishly long, okay? The endomycium, it's not actually shown here. Okay, it's shown right here. It's what's going to wrap around one muscle fiber, okay? So it's going to wrap around it. Once you get a bundle of these little muscle fibers, you have the perimycium that's going to wrap the whole thing. And then to cover the entire muscle fiber, you have the epimycium. All right. So you've seen that if you've like cooked chicken, as you're pulling around that. Yeah. What else? Anything else here? We've got blood vessels through here. We've got connective tissue. We've got tendon here. It's attaching this muscle to bone. Yeah. I feel like there's something else I was going to show you. Yeah. I think that's it. Okay. Concerns? The epimycium typically blends into connective tissue, right? So this outside covering that then leads into connective tissue. Tendons are basically cords that they're pretty tense. They're basically collagen. Collagen shows up a lot. It's this protein. It's tough. It shows up a bunch. Tendons are going to be strong, tough connectors. They're often going to be the connection that crosses a joint, because they're little. But so that's going to be like, we're going to tie this muscle onto bone, right, with connective tissue. You also have like sometimes you want the muscle to be attached with like a sheet, right? You're not tying onto anything, but you're like attaching it to, say, connective tissue, or there's cartilage, or there's bone. You're doing like a long sheet. It's called an aponeurosis. It's just the same deal as connective tissue, but instead of that tough like rope idea, you just have like a sheet. So far, so good. And briefly, I need better pictures. I don't know. I go back and forth between having these be too floofy and not floofy enough. But you can tie muscles onto bones. You can tie muscles onto cartilage, right? And you can attach it to connective to just connective tissue in general, right? Muscles can stick to all sorts of things. We're good? It's kind of just skeletal muscle in general. Looking at smooth muscle? All right. I amuse myself. Okay. Smooth muscle's not striped. I know, right? I'm hilarious. Not really. I'm not hilarious at all. But I try to not have these because otherwise it's just words. People have good biology jokes. Suggest them. I have spent more time looking up biology jokes than I really meant to. And they're really lame. Like, painfully lame. They're like, I think pitched for like 5th graders, and they're just not good. So yes, help me out if you have some. Okay. Smooth muscle, I'm not touching that mouse. Okay. Isn't stripy. They're still pretty long cells. You can call them muscle fibers. Muscle fibers, people will probably think you're talking about skeletal muscle though. But smooth muscle is that nice long shape. It doesn't have, it still only gets one nucleus. So skeletal muscle, you can put tons. Smooth muscle, you still just get one. Still a big cell though. This one's involuntary. So it's weird. Like your skeletal muscle, you're expecting to think about it and under conscious control move something. And you don't have to think that hard, right? Like to stand up, you don't have to, hopefully you don't have to like focus on standing up. But it is under conscious control. You can stop doing that if you want to. The nervous system is bossing it around. Smooth muscle, the nervous system doesn't have to boss around. So like, to have digestion happen, it can just kind of do its own thing, right? That being said, can you influence smooth muscle activity? So, like, for instance, there's smooth muscle in the stomach. Have you ever been, you don't have to, maybe, have you known somebody who's been, like, they just broke up with their significant other, or they're about to take the one single test that will determine their entire life fate or whatever, right? Is it hard? People often feel weird with their digestive system during this time. Like what people are thinking about can't influence smooth muscle, all right? But that's through a long series of events. Basically, it's how stressed you are, right? But smooth muscle isn't under conscious control. People can influence what they're thinking about and how they're doing, breathing, that kind of thing. But you can't just like think and be like, okay, stomach churn faster. People can't do that. And it's weird to me, but it totally can. It can contract without outside stimulation. You're gonna see smooth muscle on hollow organs on the inside. So you've got the stomach, the urinary bladder, respiratory passages. Quick question, just because I think it's interesting. Smooth muscle's involuntary. The urinary bladder is involuntary. How come? You could hopefully control when you pee. Yeah.
So like you could decide whenever you like actually go.
Yeah, she's right. So the bladder part is involuntary, but you've got a voluntary sphincter that you can be like, nope. Which is also why you could like condition very small children to pee. Like people do this. Have you ever heard about this? People like condition kids to pee on like a whistle. It's a thing. Okay, just people are looking at me. Yes. It's more of a thing if you're using non-disposable diapers. Apparently, so my sister-in-law is Chinese, and she's like, yeah, everybody did that. Just like you hold the kid over and you're like whistle, and they pee, and you just associate it. My sister tried it with her first kid, but then gave it up with her second because she's like, it's too much work. Anyways, but you can't actually like have very small children know when they need to pee because they don't have the muscle control yet, and it's just involuntary. But by the time you get older, you figure this out, so that's good. But yeah, this is all this to say involuntary. And you're seeing in the hollow organs. You'll see it something like this. So we're looking down. We've got mucosa. That's great. We care about these rings right here in the hollow organs. And you'll note how there's the smooth muscles set up in a couple different ways. And in the stomach, it's really noticeable. It's set up in several different orientations. But you've got this circular layer where the smooth muscles pointing around this way. So when it contracts, it squishes in. And you've got it also in this longitudinal layer where they're oriented the other way. So when this squishes, so when this contracts, it's going to get smaller, right? When this contracts, what's going to happen? It's just somewhere in the intestines, right? So you can either squish it down or you can push stuff along, right? You can move things in two different directions. In the stomach, you actually do like all three. We're okay with this idea? Yeah, smooth muscle is kind of cool. And is getting more research attention now. For a long time, it was like skeletal muscle, that's what we care about. Now it's like smooth muscle's kind of interesting. It's doing things on its own. Also, cardiac muscle probably has the most research, where it's like, oh yeah, really, really needs to happen. Remind me where cardiac muscle is? Heart. Anywhere else? Nope, just the heart. Uh-oh. Yep. Okay, so cardiac muscle usually has a single nucleus. Sometimes might have more, but typically just has one. It's also stripy, so in some ways it looks like skeletal muscle, except it doesn't have these really, really long cells. It's got a lot shorter cells, and they branch instead of being straight. They're very connected. So like all muscles are going to be kind of sort of connected to their neighbors. Cardiac muscles are joined with something that's called an intercalated disc, meaning they're basically open to the other cell. Like two different cells, they've got separate plasma membranes, but they've got connections between each cell. So when we get to the heart, this becomes really important because as one cell changes electrically, the other cell notices it in real time. It doesn't have to like send a message, it just feels what the other cell feels. Yeah? The heart is kind of halfway between smooth muscle and skeletal muscle in that it, like skeletal muscle, can just contract on its own. The heart has a pacemaker inside, not like a pacemaker that was medically put in. It has its own internal pacemaker, right? Just like goes along. And that's just a steady rate. But the nervous system also talks to the heart and can adjust that rate. Is everyone with me here? And similarly to how smooth muscle's not under conscious control, cardiac muscle's not under conscious control either. But again, what you're thinking, what you're doing, you can adjust this. So like, if you hold your breath, you can change your heart rate, right? Also, if you just like think calming thoughts, you can control your heart rate. Some people are way better at this than others, but you can get better at it if you try. You can also, if you remember that jerk who stole your lunch money like 4 years ago or 8 years ago, however long ago, and you haven't gotten over it, and you just ooh them, you can also just think about it and raise your heart rate, if you want to. Questions? Okay. It comes up in these bundles here in the heart, and it's gonna just squeeze. We're spending like an entire week and a half on the heart, so you'll see it. Okay, we're gonna now that we've seen those three types, right? We've got skeletal, we've got smooth and cardiac. We're gonna talk about skeletal muscle for like the rest of today. Yeah, is everyone okay with this? For all I was like, it's the least interesting type. It's it's the classic type. And we're going to watch how it contracts, because it's important. Okay, so skeletal muscle is going to do several things. It's going to be producing movement, that's the classic one, right? Like, move. It's going to be holding posture, right? So you don't, like, droop. Has anybody ever, I guess, has anyone seen someone with stroke, like in their face? What happens?
One side is different, you know.
Yeah, like one side's not doing its job, right? And like there's also often drooping, right? And it reminds one, when you see it, like, oh man, that's rough. But it reminds one that even if you're not thinking about where to keep the muscles in your face, you're holding your face in a specific way, right? It's skeletal muscle there, right? But just maintaining your lips where they're supposed to be, you don't have to think about it, but it's under conscious control. Everyone's okay with that? You can just maintain a posture. Skeletal muscle also stabilizes joints. So, yeah, it's going to keep you from, like, twisting your ankle as badly as you would otherwise. And it's going to generate heat. Typically, and so, is anybody locked in a constant struggle for what temperature a room should be? Roommates, no? Oh man, you people, like, deal well with your interpersonal problems. Once upon a time, sharing an apartment with two other people, right, so there's three of us. And, like, I was right in the middle, but one of my roommates, every time she walked past the thermostat, like, every time she'd turn it up. And the other roommate, every time she walked past the thermostat, would turn it down. And did they, like, talk about their feelings? No, they just both, like, constantly push the buttons. Anyways. Typically, people with more muscle feel warmer. You might have noticed this, right? And with less muscle are going to feel colder, just because muscles generate heat. It's not always, but that's the general idea. Question so far? Okay, so that's big scale. We're going to look small scale. Oh. Okay, skeletal muscle. We're going to look at skeletal muscle, and it's a bizarre cell, because in a lot of ways it looks like a neuron. When we start seeing neurons, it's going to look similar to that. It's going to do stuff with electricity, and that's not normal. Most cells don't really care about, like, zappiness, but muscle cells do. It's also really big, and it's all over the place, and it's going to have a whole bunch of fibers. So in some ways it looks like something like fibrocartilage, but it's not at all. Stay. Okay. We've got what's called the sarcolemma. Everything in muscle cells, it's like a normal cell, but they all get their own names. Okay? So, because they're special. So, the plasma membrane, you'd normally just call plasma membrane, right? Edge of a cell. This one's going to be called the sarcolemma. Sarco for flesh. It's a plasma membrane that's extra special for muscle cells. We're good? In a normal cell, you've got a couple organelles. You've got, like, mitochondria, you've got the Golgi, you've got the nucleus, right? Muscle cells get these organelles called myofibrils. They're going to be really, really long, and that's what, inside that you've got different bands, and that's what's going to make it look stripy. We'll see pictures if this changes. And push the button. There we go. Okay, so we've got the sarcolemma here. We've got a myofibril here. Question, this might be dumb, but is this a cell, and then this is a whole group of cells? There's a really common misconception. They're like, hey, look, this thing looks like a cell, and you have a whole bunch of them, they're together. No, this is all one cell. This is all one cell. Okay? And I'm with you. I see why you want to be lots of cells. This is one cell. This sarcolemma is what normally would be a plasma membrane. Okay? And these myofibrils, got a whole bunch of organelles packed into this one freaky looking cell. And you've got lots of these different, you've got a nucleus here, you've got a nucleus here, you've got lots of different nuclei. Is everyone okay with this picture? And can we all appreciate for a moment that it looks weird? This is not what a normal cell looks like, but this is a normal muscle fiber cell. Yeah? So at some point, I'm going to ask you some question. I think I have in lab today too. Is this a muscle cell? This little myofibril? And the answer is what? No, absolutely not. Haha, of course not, right? We're good? But I get why you want it because it's an organelle, right? But it's big. Okay. So, lots of terms today. It's not terrible. And mnemonics are helpful for you. We're gonna look at it right here. We're going down from this big picture. Oh, sorry, let me be coordinated. From this big picture, we're gonna go look at a myofibril now. We're gonna go in. And we've got a couple different portions. Come on, change. There we go. So this thing's running long. We don't show it all the way. I need my stick. I really kind of like this room, by the way. It's got like random crap for Gen Bio and all sorts of things, and nobody else uses that, so I'm all excited because I can just leave stuff. Okay, so we've got this little stripy bit here, right? Like fibers there. We've got lots and lots of fibers running long ways, right? We see that some are really fat and some are really thin. Okay. All of these portions have different names. So right here, this is called the M line. It's what's called the center of the H zone. The H zone is the portion that looks lighter. That when you're looking at the tissue, it looks lighter because there aren't all these filaments in there. The I band, sorry, the L band, I just called this right. This light right here, it doesn't have any of these fat, chubby fibers, right? A whole sarcomere is from one Z disc to the other. The Z disc is right here. It looks zigzaggy. It's got only thin filaments in it. There's terms for everything. Yeah. The A band's the dark band, right there. It's where you see a bunch of, you see all the fibers. Yeah? So far, so good? You'll see it's a repeating pattern. It goes Z disc, right? This zigzag here, that's got actin. Actin's really, really thin. Actin, when you looked at it in like Gen Bio or other biology things, actin was what you called, microfilaments, or so thin filaments. Here we're calling it actin. Myosin's nice and thick, so you've got actin here. Myosin's nice and thick. Myosin's attached here to the H zone. And this pattern just repeats over and over and over. We'll see pictures here in a minute. All right. This between Z disc and Z disc is called sarcomere. That's the unit that can actually contract. All right. It's worth remembering that the thick filaments are myosin and the thin filaments are actin. And actin and myosin are going to interact together, and it's going to be important. All right, so the thick filaments, they're myosin. They may have a protein called myosin. They've got a whole bunch of ATPase enzymes, and they've got what are called myosin heads. We'll see pictures. These heads are going to attach to thin filaments, and when you've got the head attached and you've got the thick filament, thin filament, it's called the cross bridge. We'll see a picture. We'll see a video. I have random things for online people, lots of study stuff. Watch the videos. I've got videos. And the thin ones are going to be actin. Wait, video. Picture. You got this picture? Right? This thick one, remind me what the thick one is? Yeah, and the thin one? Beautiful. And they're going to form cross bridges. And this whole thing's the sarcomere. Okay. What? All right. Okay. Just things to know, and then we'll actually see them doing stuff in a second. The sarcoplasmic reticulum, the SR, again, the names are funny. Remember how back up here we have the sarcolemma, special plasma membrane? Now we've got the sarcoplasmic reticulum, which is a fancy version of the endoplasmic reticulum. So in a normal cell you have the ER, in the muscle cell you have the SR, okay? It's gonna be holding and dumping out calcium. Calcium is hugely important. So you know how like, oh yeah, by the way, good job on the exam, everybody. Feels like a long time ago. People did really, really well. Yeah, I was impressed. So go you. Was there anything I want to say about that? Yeah, no, people did a really good job. I miskeyed one on the lab exam, and the first person who checked the grade was clever about it. Was like, you miskeyed, I just keyed it backwards. I'm like, all right, so I fixed it probably before most of you saw it. But I think that did. That was the problem. Oh yeah, you might have also noticed one of the problems was there was an answer that didn't actually have a question. It was like, I had like a picture with all the little things. It's like A, B, C, D, E, F. And it only goes to like D on the picture, but that's okay because the answer was like A or something. So yeah, just got excited about giving you options. I like online tests only because I get to see like which question is hard, which question is easy. That is cool. But people did a really good job. Right. Where's it going? Oh, right. In the test, people were talking about bones and how bones act as the calcium reservoir for the body. This is one of the reasons this is hugely important. Skeletal muscles in general, have got to have calcium to be able to function. And the SR is going to hold this. It's gonna hold and then release this calcium. All right. Cool. A couple more terms. We're nearly there, and then we'll see it in action. But it helps to have a few words. What we've already seen them today. Thank you for your beautiful explanation. There are a couple things that muscle cells do. All muscle cells are going to be irritable. That's really what it's called, irritability. You could also call it responsiveness, which is just basically if you poke them, they respond. It's not really poking, but if you get a stimulus is sent, they'll respond to it. So something like a red blood cell, you could, throw a neurotransmitter at it and it wouldn't notice it. It would just kind of be like, yeah, okay. Muscle cells, you throw a neurotransmitter at it and it notices. Okay, so it's responsive. It's irritable. I don't know why irritable is the term that's used. I feel like responsiveness is a better term for it, but that's okay. They also all are going to be contractile. They have contractility. That means if they get the right stimulus, they can shorten up. Okay, so they can contract. So a muscle cell can contract. What happens once it's done contracting? What do you want it to be able to do? You want to be able to go back, right? Did anybody as a child ever play with those little finger trap puzzles? It's a little tube, you stick your fingers in, you can get them in, and then you're like, right? Okay, you can't get them out. I realize you've gotten them out because you're here. Congratulations. But you need it to be able to undo. So this next one, extensibility is, nope, wrong way. I put them in the wrong order, sorry. Elasticity is the after, okay. You need to be able to contract, you need to be able to extend, right? So you can contract, you can then also get it out long. And then you need it to be able to bounce back to normal. So we've got three things going on here. Okay. First one, it can respond. It can respond great. It can shorten, it can stretch, and it can bounce back to just the normal shape. Yeah? Questions about that? Okay. I'm just going to show you a video in just a sec. Here we go. Last thing before you see video. Has anybody heard of the story of Frankenstein? Read it, watched it, right? This is Dr. Frankenstein who, using mostly electricity and dead body parts, puts puts a monster back together, zaps him with electricity, and it's alive, right? Okay, this story, it's interesting, right? It's not like biologically relevant particularly. It's interesting though because at the time Mary Shelley wrote it, yeah, Shelley was Frankenstein, right? Who's Dracula? Stoker. Yeah, okay, thanks. Yeah. Mary Shelley wrote Frankenstein. At the time, muscles were just being figured out, right? And people were realizing that, like, this electricity stuff is really cool, right? Electricity is inherently interesting. But if you put electricity on muscles, they move. And even if you put electricity on, like, dead muscles, they move. I mean, if it's not, like, too dead, right? And people are like, maybe electricity is what life is, right? Like, maybe somehow we're just generating electricity and, like, doing stuff. And so if we can, like, you know, run electricity back in on tissue, we can have Frankenstein, which is maybe a little bit traumatic, but there's something to be said for that. Like, if you could run a pacemaker in on the heart, right, you can make it keep going. And the ability for the nervous system to send signals, electrical signals, and for muscles to handle electricity, I mean, it's also handling neurotransmitters, is really phenomenal. And there was something to this. They're like, yeah, muscles care about electricity, and they do stuff. But yeah, what's going to happen is a skeletal muscle is going to be stimulated to contract. A nerve cell, a motor, it's called a motor neuron. It's just a version of a neuron. It's going to go and it's going to signal the muscle. It's going to say go, okay? And when it contracts, interesting things happen. The motor neuron coming in can signal just one skeletal muscle cell. Typically, it signals a bunch of them. So in a picture like this, we've got this motor neuron coming in, and this same one is going to signal a whole bunch of different muscle cells. So one signal can signal a whole bunch of different muscle cells. And this, I realize, this picture, it's kind of a rough picture because it's at many different magnifications, muscle cells look similar, right? So if you're looking just at one cell and you're seeing all the myofibrils, it looks very similar to when you've gone out and you're seeing a whole bunch of muscle fibers together. Here's another picture. We've got all these different muscle fibers, so all these different muscle cells. These are different cells, right? So this cell right here might have a couple different nuclei in it, but it's all one cell, this long fiber. And we've got a neuron coming in, and you see all these little branching spots? That's where that neuron is touching the cell, and it's going to send signals. Actually, these aren't neurons here that we're seeing. We're seeing the neuromuscular junction. I've got a lot of words, mostly for online people. Hi, online people. We're going to watch a video here to see stuff. It's also embedded in here. It's embedded here. So the slides go through like everything, boom, 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 boom. It's way easier to see it. But before we do that, I wouldn't want to make it like too interesting. Everybody pick a number. Logan, can start with you. Pick a number between 1 and 7. Just start off with 1 and we're going to go and everybody's going to get 1. Say it loud. 1. Cameron. Shaylee. Kimmy. You're 4. 5. 6. 7. 1, 2, 3, 4. Okay. Everybody have a number? Know the number? This film is going to yap about stuff. It's going to be like, the first step, blah, 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 blah. The second step, blah, 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 blah. Okay? So we're going to watch it, and then we're going to see if we can recreate the steps with a dramatic interpretation. Yeah? Everybody has a number? Know what's going on? Online people, just watch the video and feel involved. The person who narrates these is so calm, which is good. It's good to be calm.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber.
This was not the one I was wanting to show you. Just a second. Is this the same one? Yeah. Oh yeah, no, this is the right one, sorry. I watched it on like triple speed and I forgot the first part, sorry. It does start labeling things 1, 2, 3. Sorry. I get nervous.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps.
Here we go.
Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay. There's a lot going on there. I recognize there's a lot going on there. And there are a couple spots that people get tripped up by. So, we're going to talk about stuff. Let's cut here. We're going to talk about stuff and see how we're doing. So, people number 1, what happens first? So I'll catch it, whoever's first one. Yeah.
Was it impulse?
Yeah, an impulse comes down. It's called an action potential. It's coming down the neuron, right? Whoever else was one. Okay, so okay there. That's an action potential, right? Yeah, we're okay with this. It's an action potential of the neuron. We haven't hit the muscle yet. Okay. This is going to become important because we're going to have an action potential in the muscle cell in a minute. All right. So this is an action potential. It's an electrical signal. Action potentials are electrical signals. Electrical signal going from the neuron down to the very, very end. Okay, so it's going to go down to what's called the neuromuscular junction. So it's where the neuron ends, and we've got the muscle fiber right next to it. Okay, and action potential coming along. Okay, what happens when it gets to the end of the neuron? Person number 2, did you catch what it's called?
Calcium ions moving.
Yeah, you open these voltage gated calcium channels. Okay, so voltage gated means the voltage changes them. Okay, so we're here. I'm just gonna act it out because hi online people, it's probably easier than the board. Okay, so action potentials come down. This electrical signal. When this electrical signal hits the end there, gets the end, it opens calcium. It opens ion channels that let calcium come in. Right? Great. So it's voltage gated calcium channel. Calcium then comes in. Whee! Right? Okay. Person 3. When calcium comes in, what does that make the neuron do? Yeah. Yes, absolutely. Okay. Is everyone still with us here? We've had an action potential, come down. The action potential is electric, right? That electricity's open these gates for calcium. When calcium comes in, calcium then dumps out acetylcholine. So there's these bags of acetylcholine, right? These vesicles of acetylcholine. When calcium comes in, a series of steps, it's complicated, but a series of steps, it's going to let these vesicles fuse to the membrane and pour acetylcholine out. Okay? Right? So the neuron wasn't sending acetylcholine down the length of it, right? Nope. It's already there packaged, but calcium coming in lets acetylcholine out. Everyone's with me here? Cool. Okay. So acetylcholine's now what's called the synaptic cleft, because it was right there. There was a neuron, a little tiny space, and then the muscle cells right next to it. So right now, we dumped acetylcholine out, and it's chilling just right between them, and the space is microscopic. What happens next? I guess it was 4. Anybody remember? Yeah. Okay. Yes, it's gonna bind to ion channels again. We're gonna see pictures again here, but we're gonna see talk through the whole thing just for one. How are we doing? Okay. So acetylcholine's been dumped out of the neuron, right, into the synaptic cleft. It goes over here and it binds to ion channels. These are ligand-gated ion channels. We had ion channels a minute ago. What kind of ion channels did we have a minute ago? Voltage gate. A minute ago, electricity turned them on and opened them, right? These ones here on the muscle cell, acetylcholine is the key, okay? So it's not electricity there that's turning it on. It's acetylcholine that turns it on. Acetylcholine is not pouring into the muscle cell. It is not entering the muscle cell. Nope, does not do that. It shows up as the key to the door, and you just kind of leave it there, and eventually you're going to pull it out. But that's going to open the door and let other things through. Everyone's with me here? Acetylcholine had been the neuron, went across the synaptic cleft, binds to these ligand-gated ion channels, and these ligand-gated ion channels then open. Person 5, I honestly can't remember who you are, I'm sorry. Oh, hi. What ions move?
Sodium moves in and then potassium moves out.
Yes. Okay. Here, this is where stuff's actually moving into the muscle cell or out of the muscle cell, right? Sodium is going to, tell me again? Moving the cell. And it's going to pour in fast. Okay. Sodium's got a positive charge. All right. And sodium is just going to rush into the cell. Whee! Really, really fast. Okay. And like she said, potassium's going to move out, but potassium's kind of slow. Who cares about it right now? We'll care about it in a second. But, sodium's going to rush in. Oh yeah, person 6, I think I stole yours. Anything else you want to say about that? Yeah, you're gonna have positive and negative charges. Well, you're gonna have positive charges moving, and you suddenly got a lot of positive charges inside relative to what you just barely had. We're okay with that so far? Hi. I think someone's looking for something. Okay, great. And then person 7, once that happens, what happens?
Once it reaches its full potential, it lights into that cycle.
Yeah, you get an action potential, right?
Yeah.
Once you hit a certain voltage, I'll get to it. Once you get a certain voltage, you have an action potential. We had an action potential already, though, right? Where do we have an action potential? In the neuron, right? It was an electrical signal, but now we've got an electrical signal in the muscle fiber. Is everyone cool with this? There's some parts that are easy to get jumbled up because you're like, action potential. We're talking about 2 of them. We're talking about a neuron action potential and a muscle fiber action potential, right? Ion channels, we're talking about the voltage-gated calcium channels that are on the neuron. They're going to let calcium in to dump acetylcholine out. And then we have acetylcholine channels that are going to let sodium and potassium cross. So far, so good? Okay. I recognize a lot going on here. Okay. This is all just the words for this. We're going to have calcium channels open. This is after the nerves. Did I skip anything? All right, I just wanted to tell you a couple words, sorry. Neuromuscular junction, where those 2 attach. Neurotransmitter, it's this chemical that the nerve gets to release. Acetylcholine is the one that stimulates skeletal muscle. Okay, acetylcholine's abbreviated ACH for acetylcholine. Great. And then the synaptic cleft is that gap between the nerve and the muscle. It's a tiny gap, but it's there. They don't actually touch, then they're not physically in contact. There's a tiny space. And it's just got interstitial fluid in the middle of it. Okay, so here we are. Same steps, you all went through it, but just to see them here. Calcium channels open. Calcium in the neuron. Calcium's going to have acetylcholine dump out. Acetylcholine wanders, diffuses across that gap, attaches to the receptors, does not enter. Oh my goodness, people desperately want acetylcholine to enter neuron muscle cells. It doesn't. Okay? If enough acetylcholine is released, these ion channels open and the cell is able to become way more permeable to sodium. And so sodium rushes in, potassium leaves. The cell is then what's called depolarized. The cell was polarized before. The muscle cell was polarized before because it was held at a fairly negative potential. The voltage is fairly negative. So it's depolarized because positive charges are coming in, so it's bringing it less far from 0, right? It's making it polarized. That depolarization, we had the cell suddenly become slightly more permeable to sodium. As that happens, you can open more sodium channels. So if you have voltage-gated sodium channels, the more positive charges you have coming in, the easier it is to open those up, right? Once you get that action potential, the action potential in the muscle, you're committed. Okay, so if you just have a little bit of acetylcholine and you don't have enough to make an action potential, meh, not a big deal. Once you have enough sodium channels open for an action potential, that muscle cell is contracting. It's committed. It's kind of like a light switch almost. Like, sorry, I'll stay here. You can flip, you can turn the light switch, turn light switch, turn light switch, but once you click it past a certain point, the light is completely on. You can't have the light be like partially on. You can have it be on or off, and once you cross that threshold, you're done. Questions? Okay. And then finally, acetylcholine esterase is going to eat up acetylcholine, and then the muscle contraction is done. And some of the acetylcholine diffuses away as well. So far, so good? Lots of parts. The cell then is going to go back to just chilling once, potassium ions are going to diffuse out the cell, and the sodium-potassium pump, the sodium-potassium ATPase is what it's called, It just keeps pumping sodium and potassium around, and it's going to go back to their original positions, where there's a lot of sodium outside. This pumps sodium out and pumps potassium in, because then when the signal happens, sodium comes in, potassium goes out. This poor little pump just keeps shoveling things all the time. Lots of pictures here. Is it helpful to walk through the pictures? Should we walk through the pictures? Okay. Is this big enough? Can you see it okay? Okay. This whole thing, right? This is the neuromuscular junction. This is the neural part. This is the muscular part. And it's the junction. It's both of them. Okay. This nerve impulse came down. Whee! Right? Got an electrical signal here. That electrical signal opens these calcium channels, right? There's this channel here. It lets calcium in, right? Once the voltage reaches, calcium comes in. This calcium comes in. Now suddenly you've got calcium at the edge. The calcium makes it so that these acetylcholine vesicles fuse here and dump out. Makes this exocytosis happen. All right? You can see it here. Acetylcholine then that just got dumped out, tiny and green here. It diffuses across this membrane, across the synaptic cleft, and there are these little. They're shown in purple here. These receptors that notice acetylcholine. And when acetylcholine hits it, they change shape and they open. And then this acetylcholine here in green opens up this channel. This channel has sodium and potassium move, and sodium can move faster than potassium can move. So sodium is rushing in, potassium's trying to rush out, but sodium beats it on the race, right? They're both moving, but more sodium's coming in. So sodium is going to come in, and this suddenly is going to make this more positive, which is going to lead to the action potential, right? So suddenly you have a ton of sodium inside, a lot of positive charges, right? And then this channel here was being held open by sodium. Right, it was holding it open. Sorry, acetylcholine was holding open the channel, letting sodium and potassium move. Sometimes that acetylcholine just like squiggles off, just because there's random motion associated with all molecules. So sometimes it just wiggles off. But more often, how it goes away is acetylcholine esterase, which sees this stuff and likes to eat it up, comes and it breaks it down. As acetylcholine gets broken down, these channels that were being held open to let these ions pass close back up, and then you're done. We're good? Questions about this? Okay. We're supposed to end at 40, right? Okay, so I think I'm gonna yap for another couple minutes. I had these plans about that we were also going. So this is what happens at the neuromuscular junction. I had these plans like, and then we'll figure out how like the cross bridge cycle works and blah blah blah. I don't think we're gonna do that today. We'll see it in lab. You'll see it in lab. Come in for today's lab if you're not ill, right? I've got some muscle fibers that we're gonna put under microscopes and throw ATP on and watch them squish. I mean, it's cool. You kind of have to be like watching for the cool thing to happen, because otherwise you're like, I don't notice it. But it's kind of cool. But okay, your book has this analogy here that's like a fire. It's basically once that signal goes, once the action potential happens, it's going fast in both directions along the whole sarcolemma, the whole plasma membrane of this muscle fiber, this muscle cell. All right. Okay. Just a sec. Do we want to see this? Okay, we're gonna watch it. You don't need to memorize it. We're gonna watch it just so it makes sense for today's lab, but then we'll spend a fair bit of time on Thursday talking about this, okay? Because once we're once we're here, yeah, the action potential happened. What does this mean? What happens here? So don't worry about adopting one of these personally. Let me show it big, actually. Does having these embedded, does it completely make it impossible to download them? Is the file size too big for you guys on Element? I hope not. Online people, we're watching this
The
video.
contraction of a skeletal muscle generates.
Loud enough?
The force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, I realize that it doesn't make a ton of sense right now because we need to go over a bunch of things and we kind of miss a step between like, yeah, we have an action potential happens and then all this. But it'll help for labs today to be like, oh yeah, there's going to be some sort of binding and contraction. Okay, so again, we'll talk about this on Thursday. I'll see you in lab. Online people, I will send you your lab. Yeah, we're good?